This is part C of chapter 3 on the cells, the living units. And this is the second part of a relatively lengthy chapter. And this one is focusing on the cytoplasm. So all cellular material that is located between the plasma membrane and nucleus. This is composed of the cytosol, which is a gel-like solute or solution made up of water and soluble molecules such as proteins, salts, sugars, etc. Then we have inclusions. These are insoluble molecules and they vary with cell type. Examples include glycogen granules, which is how we store sugar in cells, pigments, lipid droplets, vacuoles, and crystals, and then organelles. And the organelles are the metabolic machinery, structures of the cell, each with specialized function, and they're either membranous or non-membranous. So there's some that have a bilayer membrane and then those that do not. So in 3.7 we're going to be looking at the cytoplasmic organelles. So our learning objectives here are to discuss the structure and function of the mitochondria, and then discuss the structure and function of ribosomes, the endoplasmic reticulum, Golgi apparatus, including functional interrelationships among those organelles, and then compare function of lysosomes and peroxisomes, and name and describe the structure and function of cytoskeletal elements. So as far as the organelles go, they're illustrated in the image on the right. And they include the ones that are membrane-bound organelles. They include the mitochondria, endoplasmic reticulum, or reticula plural, Golgi apparatus, peroxisomes, and lysosomes. And then there's the non-membrane or non-membranous ones, including ribosomes, cytoskeleton, and centrioles. As far as the membranes go, the membranes allow for compartmentalization, which is crucial to cell functioning. What that means is you can separate metabolic processes within the cell. And so we're going to explore every one of these organelles. Take some time if you can. You do have some, and recognize each of these structures, not just memorized in this illustration, but what their structural features are, so that in any drawing representation, three-dimensional model that you find in the lab, you can distinguish one organelle from the other. Mitochondria is going to be the first organelle we're going to look at. It's membrane bound. Sometimes they call it the power plant or powerhouse of the cell because it's where they produce the cell's energy in the form of ATP, and this is done by aerobic or oxygen requiring process called cellular respiration. So in this process, food is oxidized, and the energy from the electrons in those food molecules is going to be used or harnessed within the mitochondria to take ADP and reattach an inorganic phosphate to it, and that would form our the main molecule we use as energy currency in the cell. The mitochondria itself is a double membrane, and that inner membrane is going to have folds called cristae or crista singular. So you can see in our image here on the right, there's two membranes. There's an outer one and the inner one is folded in there. And within those folds or those cristae are embedded proteins that play a big role in cellular respiration. And that entire process will be covered in an entire cell on cell energetics, which would be covered later on in your textbook. The mitochondria also are going to have their own DNA, RNA, and ribosomes. So there's interesting theory on how the mitochondrion came to be from simpler cells billions of years ago, but no time to get into that. But that having its own DNA and ribosomes is part of the evidence for that process. The mitochondria do resemble bacteria, and they are capable of multiplying within the cell on their own, just as bacteria do, which gives an idea of the hypothesized origin of mitochondria. The process that bacteria and mitochondria go through to multiply is called binary fission, which just means split in two. And then we have ribosomes, and these ribosomes are non-membranous organelles, and they're the site of where protein synthesis occurs. So these are the protein factories. The ribosomes are going to be made up of protein and a kind of RNA called ribosomal RNA. There's two switchable forms found in the cell. One is free ribosomes, and the free ribosomes you can see as these red dots in the diagram on the right. And they're floating freely. And this is the site of synthesis of soluble proteins that are going to function in the cytosol or within other organelles. So here we might make proteins that are going to be part of the metabolism occurring within the cytosol itself. And then we have the membrane-bound ribosomes. Those are going to be attached to the endoplasmic reticulum, and if so, we're going to call that the rough endoplasmic reticulum. And here, because they do produce proteins, here we're going to be making proteins that would be have some various functions. They might be incorporated into the membrane. They might become lysosomes, which are bodies that do digestion, or they may be exported from the cells secreted outward. So if we look here on the right, and I zoom in, you can see the membrane that's folded, this blue one here, studded with ribosomes. Those are going to be those attached ribosomes, the membrane-bound ones on the rough endoplasmic reticulum. And then you have free ribosomes in the cytosol there, but both basically have the function of producing proteins. And then we have the endoplasmic reticulum, and the endoplasmic reticulum is in blue in the picture on the right, and there's going to be two kinds. And these are going to be a series of parallel interconnected cisterns, which are going to be flattened membrane tubes, and they enclose fluid-filled interiors. And this is going to be where certain parts of your cell metabolism take place that are kept separate from the rest of the cytoplasm. The endoplasmic reticulum is going to be continuous and extends outward from the nuclear envelope. So the nuclear membrane actually has two membranes, and the inner one, the outer one, folds outward to become the endoplasmic reticulum. And there's going to be two kinds. If you have ribosomes on there, it's a rough ER. With no ribosomes, it's a smooth ER. So let's just zoom in quickly here, and you can see if I point to the nucleus here and we start working our way from inside the nucleus, the first membrane you come across is the inner membrane, and then you have the outer membrane. That outer membrane extends outward and basically becomes the endoplasmic reticulum. So we have our our rough endoplasmic reticulum with proteins, and then as we extend further out, we transition into the smooth endoplasmic reticulum. It tends to be a bit more tubular in nature. So looking at the rough ER, what makes it appear rough gives us an idea of what's going to go on in there. It's rough because it's going to be studded with ribosomes that are attached. So if we have ribosomes, then we know that the job of the rough ER is to synthesize proteins. And for the most part, these proteins are going to be kept separate from the rest of the cytoplasm. So one possibility is that those proteins could be exported or secreted from the cell. In other cases, we may be synthesizing proteins that might become part of the plasma membrane. We know that the plasma membrane has proteins. They have to be made somewhere, so they're made in the rough ER and then placed in the plasma membrane for their functioning. Now the proteins, when they're being made, they're going to enter the cisterns or the spaces within the ER, and then they can be modified as they go through these fluid-filled tubes of the ER. The final product is then going to be enclosed in a transport vesicle, and then that transport vesicle buds off the ER, and it's going to make its way to the Golgi apparatus, and there in the Golgi apparatus it would be further modified. So we'll talk about the Golgi apparatus in a little bit, but basically any product produced in the rough ER would end up in the Golgi for further processing. And then looking at the smooth ER, so the smooth ER, so we're coming off the nuclear envelope right here, and then here's the rough ER. If you go further out, then you run into a more tubular smooth ER. And so what the smooth ER is going to be a network of loop tubules, and again, continuous with the rough ER. That means extending from the rough endoplasmic reticulum. The enzymes found within its membrane, so it does have a plasma membrane too, are going to have some functions. So this gives what the function of the smooth endoplasmic reticulum would be. One is lipid metabolism. So here, cholesterol and other steroid-based hormones can be synthesized in there, and lipoproteins can be produced in there. We also have another function of absorption, synthesis, and transport of fats. Another big thing the smooth ER can do is detoxify certain chemicals, like drugs and pesticides, and it can convert glycogen into free glucose. And another big function that we see, especially within muscle, is that it's going to be a place where we store calcium, or calcium ions. When I say calcium, I'm referring to calcium ions. And in this case, it would have a special name within the muscle called the sarcoplasmic reticulum. And so it would be specialized smooth ER found in skeletal and cardiac muscle cells, where it would store the calcium. The calcium becomes an important player in the contraction of those muscle cells. Then we have the Golgi apparatus, and so when you look at diagrams of the cell, they would suggest that these flattened sacs are separate from the endoplasmic reticulum. So these are flat membranous cistern sacs, so again they have space within there. And the job of the Golgi apparatus, it modifies, concentrates, and repackages proteins and lipids that come from the endoplasmic reticulum. And 3 steps are involved here. So number 1, we're going to have transport vesicles from the endoplasmic reticulum are going to move their way and fuse on the cis face of the Golgi. The cis is going to be cis means is referring to the inner side here. So if we look at the cell model right here, the endoplasmic reticulum is about right here, so those vesicles will come and fuse on this side, and this actually means on the same side. So coming from the same side as the endoplasmic reticulum, they fuse there, and so when those vesicles come in and fuse, they're going to deliver contents to the cisterns of the Golgi. So there, those proteins or lipids are going to be moved inside, and they'll be further modified. So they'll be processed, and they'll be tagged and sorted, and then repackaged. And then, so we can think of the Golgi apparatus as a traffic director. So once we've modified those materials further, then there's 3 possibilities or 3 pathways that these that the products can take once they're released from the Golgi on the trans side. So it's on the trans side that they're released, and there's 3 possibilities that can occur. And so we're going to look at those 3 pathways of the products that are coming out of the Golgi apparatus. So this, of course, depends on what's in there. We have 3 possibilities for that final transport vesicle. So here they are. There's pathway A, then B, and C. In pathway A, we're going to produce the vesicles that come off are going to be secretory vesicles, which means they're going to go for secretion. So they may contain proteins that are going to be used outside the cell. And these vesicles will fuse with the plasma membrane. So exocytosis can occur, and we can release the content. So there you can see on the diagram right here, we see the vesicle coming off, secretory vesicle, then it comes here and it fuses with a plasma membrane and releases. So that would be a pathway. An example of this would be the pancreas cells that release insulin into the extracellular fluid, so the insulin can get in the blood supply and then be circulated around to find its target cells, so the insulin can do the work of modifying those cells, metabolic activity. In pathway B, the vesicles containing lipids are going to contain lipids or transmembrane proteins, those membranes that are found within the membranes. In this case, it's going to fuse with the plasma membrane or another organelle membrane. And so what happens here is we're going to insert those proteins or those lipids into the membrane. So in pathway B, we've produced the final products are going to become part of the plasma membrane. So you see pathway B right here. We have this vesicle coming off, and it's going to contain contents that when the vesicle comes in and fuses right here with the membrane, the contents there gets placed within that membrane. So pathway B would be the second option, where the materials get placed within the membrane for whatever functions they might have. And then pathway C, the vesicles that come off may go on to form these vesicles that stay within the cytoplasm, like a lysosome, which is going to contain digestive enzymes. So the enzymes of digestion are proteins. They started their birth within the rough endoplasmic reticulum and then went to the transport vesicle to the cis side of the Golgi, and then those proteins were modified and then packaged into a vesicle that stays in the cell. So the same might happen for the production of peroxisomes. Looking now at peroxisomes, these are going to be a membranous sac containing powerful detoxifying substances that will neutralize toxins. A category of toxins include free radicals. These are highly reactive molecules that are natural byproducts of cellular metabolism. And so when they're produced, they can cause havoc. That means they can go in and react with other things and mess up other molecules within the cell, making it toxic. There's going to be two main detoxifiers here. There's going to be oxidase, which is an enzyme that uses oxygen to convert toxins to hydrogen peroxide. Now, hydrogen peroxide itself is also toxic. So then the peroxisome also contains another enzyme called catalase, which can convert your hydrogen peroxide to water, which is, of course, less harmless. Peroxisomes also going to play a role in the breakdown and synthesis of fatty acids as well. So, looking now at lysosomes, lysosomes are also vesicle-like structures. So these are spherical as well, but they're going to contain digestive enzymes. So these are going to be hydrolases because they hydrolyze. They're hydrolytic, and that means they're going to break down larger molecules with the use of water. So these are going to be considered safe sites because they isolate potentially harmful intracellular digestion from the rest of the cell. So we keep those digestive enzymes away from everything else by keeping them within the lysosome sac. And here we might digest ingested bacteria, viruses, and toxins. For example, if a white cell takes in these pathogens, the lysosome can digest. The lysosome may also go and bind with and fuse together with worn out organelles and break those down. Lysosomes also have some metabolic functions. They break down and release glycogen. They can break down and release calcium. And intracellular release in injury causes cells to digest themselves, so a process called autolysis. So if a cell becomes completely worn out, then the lysosomes play a role in intracellular digestion. I guess we can ignore this statement here for now, and we'll come back to what that means later when we cover bone metabolism. Looking at a homeostatic imbalance 3.3 in a clinical setting, there are cases where we have lysosomal storage disease, and this results when one or more of the digestive enzymes are mutated. That means those enzymes are programmed by genes in your DNA, and if a mutation occurs there, a faulty enzyme might be produced that doesn't do its job right. So now you still produce a protein, but it's not going to be correct and won't function the way it's supposed to. So the enzyme does't function, and a disorder that can happen from this is called Tay-Sachs disease. And this is a condition in which the patient lacks a lysosomal enzyme needed to break down glycolipids in brain cells. So what happens here is those glycolipids are going to build up because of this defect, and that's going to interfere with proper nervous system functioning. This disorder seems predominant in infants of Central European Jewish descent. And the really sad part about all this is that it can cause seizures, slow mental development, mental retardation, blindness, and death of the child before the age of 5. So that's unfortunate cause of a genetic mutation for just one enzyme that occurs within those lysosomes. Then we have, overall, we've described some of these membrane organelles that are referred to as the endomembrane system. And so this consists of those membranous organelles that we've discussed all together so far, with the exception of mitochondria. So mitochondria are not part of the endomembrane system. They have a separate origin. So these include the endoplasmic reticula, the Golgi apparatus, those vesicles, lysosomes, and even the nuclear and the plasma membranes. If you think about it, they're all continuous, because if we start with the membrane of the nucleus, the nuclear envelope, it folds outwards to make the endoplasmic reticulum. And then part of that membrane buds off to produce the transport vesicle that then comes and connects with the membrane of the Golgi. So that connects those two. And then the vesicles that come off of there can go and fuse with the plasma membrane. So overall, all the membranes are connected in that way. So overall, the job of all of these membranes within the membrane system are going to work to help produce, degrade, store, and export biological molecules, and to degrade potentially harmful substances within those or because of those organelles there. Here's a animation showing one of the pathways for vesicles. So let's take a look at how that endomembrane system is connected.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
So this animation here was consistent with pathway A, which was discussed earlier. Pathway A is where we produce a secretory vesicle for export by exocytosis out of the cell. Now we're going to look at the cytoskeleton, which is non-membranous, or non-membrane bound structures. And so what is the cytoskeleton? It's an elaborate network of rods that run throughout the cytosol. There's hundreds of different kinds of proteins, link rods to each other, to other cell structures within there. These also act as sort of a support for the cells. So the analogy would be bones, ligaments, and muscle. Because some of them are involved in attachment of structures, some are involved in just support like the bone would, and some are involved in movement. So the analogy is the same as we find in a skeleton in your body. There's going to be 3 types, general types. We have microfilaments, intermediate filaments, and microtubules. And these are in order here of increasing diameter or thickness of the filaments. Looking at the first one, the microfilaments, these are going to be the thinnest of the cytoskeleton elements. They are semi-flexible strands of a protein called actin. So you can see the image here on the right. It shows they're about 7 nanometers in thickness, and it's made of 2 strands of actin subunits. Now each cell has an arrangement of strands of this kind, and they all share a common terminal web. And this is going to be a dense cross-linked network of these microfilaments attached to the cytoplasmic side of the plasma membrane. And what this does, it helps strengthen the cell surface and help to resist compression on the cell. And some of these microfilaments are going to be involved in cell motility and changes in shape of the cell during endocytosis and exocytosis, and even amoeboid movement that was mentioned in an earlier video. And then we have intermediate filaments. These are a bit thicker. You can see the image on the right has them at 10 nanometers in diameter. So these are going to be tough, insoluble, rope-like protein fibers. They're going to be composed of a tetramer, which means 4 fibrils, and those will be twisted together, resulting in one strong fiber, as you can see in the image on the right. These are going to help resist pulling forces. So these would be filaments that are going to be attached to desmosome plaques, and so they act as internal guy wires, supporting and providing strength to the desmosomes that attach one cell to another. Some are going to have special names. For example, within nerve cells are going to be called neurofilaments, and within epithelial cells they're called keratin filaments. You probably heard the protein keratin at some point when it comes to hair or skin. And so here you can see the tetramer. There's one strand-like structure there, or strong fiber. And these are going to be the most static of the cytoskeletal elements. The others tend to be rearranged more often, broken down and built back up. The intermediate filaments tend to be more stable overall. So we have microtubules, and so these microtubules are the largest ones. These are about 25 nanometers in diameter. They're going to be composed of subunits called tubulin, and there's alpha and beta tubulins. So there's 2 kinds of tubulins, and these are constantly being broken down and built back up. Most are going to radiate from a structure called the centrosome. And in animal cells, the centrosome is the general location where you find centrioles in there. And over here, we determine overall shape of the cell and the distribution of organelles with these structures. Many organelles are going to be tethered to microtubules to keep organelles in place. And many substances are moved throughout the cell by motor protons. And they're going to use the microtubules as kind of a railway, like a track on there, so motor proteins can move along the microtubules. These microtubules also become important in cell division as they pull chromosomes apart, or are involved in pulling copied chromosomes apart into 2 new cells. So looking at motor proteins, I went and found a diagram for those motor proteins, and I found 2 of them. One is called kinesin here, and the other one dynein. These are complex protein complexes, again referred to as motor proteins, and they can help in the movement of organelles and other substances around the cell. They're going to use the microtubules as tracks as they move their cargo, and these proteins are powered by ATP, so the hydrolysis of ATP will change the shape of the protein and allow it to walk along the track. And then you can see kinesin, for example, is carrying a vesicle from one side of the cell to another. So this motor protein then would be using the microtubule as a track. If you ever get a chance, look up motor protein kinesin and look up some YouTube. They'll show some animations of these things. They look pretty crazy. The way the molecular biologists view these proteins. Then we have structures called the centrosome, which I mentioned a while ago, that organizes microtubules, and the centrioles that are found there. So the centrosome literally means central body. Soma means body, and it's located near the nucleus, which is where it gets its name because it's located near the center. It is referred to as the microtubule organizing center. That's what the centrosome is. It consists of a granular matrix and centrioles, and the centrioles are a pair of barrel-shaped microtubular organelles that lie at right angles to each other. So there's an image or a drawing of them, and there's 2 of them lying at 90-degree angles. Here, a new assembly of microtubules radiate from the centrosome to the rest of the cell. Some microtubules are going to aid in cell division, as I mentioned earlier, and some form cytoskeletal tract system where those motor proteins move along. The centrioles is also going to form the basis for structures called cilia and flagella, which are going to be extensions that extend outward from the cells. So these centrioles would be the basis of those structures. So, one question about the organelles we just covered is and the cytoplasm in general is, what is the fluid component between the plasma membrane and the nuclear envelope called? Is it A, organelles, B, cytosol, C, cytoplasm, or D, inclusions? Intensely biosynthetic secretory cells, such as nervous cells, would be expected to have greater amounts of blank than other cells would. So when we say intensely biosynthetic secretory cells, we're talking about cells that are producing lots of materials, proteins, for example, that are going to be secreted outwards. So neurons do this. Are they going to have A, a lot of centrioles, B, lysosome, C, rough endoplasmic reticulum, or D, peroxisomes? Now we're going to move on to section 3.8. And 3.8 is going to look at cellular extensions. The first learning objective is to describe the role of centrioles in the formation of cilia and flagella, which were mentioned a while ago in the prior section, and to describe how two main types of cell extensions, cilia and microvilli, differ in structure and in their functions. Now, certain cells are going to have structures extending from the cell surface. They include cilia and flagella, and they include microvilli. The cilia and flagella are going to aid in the movement of the cell or of materials across the surface of the cell. So the cilia and flagella are structures that extend outwards and actually can move. And in doing so, can provide movement for the cell itself or for materials over the surface. Then we have microvilli, and those are going to be microscopic finger-like projections that extend from the surface, and their main job is going to be to increase the surface area of the cell. So looking at cilia first, these are going to be whip-like modal extensions on the surface of certain cell types, such as cells that line the respiratory tract, like your trachea. These thousands of cilia are going to work together in a sweeping motion, and they can move substances like mucus across the surface in one direction. You can see that I've provided an illustration here showing the cilia on the surface, and overall this is modeling how cilia might move. They are going to have or move because of the proteins in there, and they have the power stroke and then the recovery stroke. So in the power stroke, the cilia is fully extended and waves in one direction, and when it does, it can move material, any fluids that are at the surface, so the materials would move in that same direction. Once the power stroke is complete, the cilia must recover backwards. So what the cilia does is, underneath the fluid it was moving, it comes back before it extends outward. That way it doesn't push the fluid back the other way. So it comes back underneath the fluid and then extends back outward again during the recovery stroke. So it basically comes back underneath the fluid, and then it's now reset to go through another power stroke. Overall, repeated movements, coordinated movements of cilia, thousands of these cilia across the surface of these cells, can cause overall mucus or fluids to move across the surface in one direction. So this is what occurs in our trachea. And the mucus traps dust particles and things, and so the cilia will beat and move those trapped particles of mucus upwards, and then we can swallow any of those particles, and eventually gets eliminated from our body with food waste from our digestive system with our feces. And then we have flagella. So flagella are going to be basically longer extensions, and that would prepare the whole cell. So the whole cell, we're talking about moving here, and here the only place you're going to find it in human biology is in the sperm cell. So the sperm cell is moved by way of a flagella. Both structures are going to be made of microtubules, and they're going to extend outward from those centrioles. And these specific centriole structures are going to be referred to as basal bodies. So they're going to look like the centrioles we just saw earlier. but they're going to extend from the base of each cilium and flagellum, or they form the base of these cilia and flagella. So if we look at the internal structure of cilia and flagella, they're going to have what's called a 9+2 microtubule pattern. And that's going to be 9 sets of double tubes surrounded by 1 pair of double tubes. As you can see the electron micrograph of a complete cross-section through there. You can see the 2 tubes in the middle, that's the +2. And then if you count all the way around, there's going to be 9 doublets. So there's your 9+2 of the internal structure of the cilia or flagella. You can see in the drawing here, where I'm drawing here on the bottom right, that would be the plasma membrane. And so inside would be the cytoplasm. Here's your basal body we mentioned earlier, which is structured just like a centriole, forms the basis of it. And then from there extends outward this 9+2 arrangement. You can see it's still surrounded by a plasma membrane, but the structure extends away from the cell. So it's that arrangement that causes, or that gives the cilia or the flagella the structure. Now, if we look at the basal body is going to have a different arrangement. It's going to have a 9+0 pattern, but except the 9 is going to be triplets with no central tubules. So we can see if we count here what a basal body looks like in cross-section, or centriole for that matter, it'll have the same structure. It's going to be 9 triplets of these microtubules there. So the central cross-section would look somewhat different, but the 9+2 tubules are going to extend outwards. Now, if you look here, the image on the right, what's going to happen here is that those tubules are going to be associated with dynein proteins, which we saw in an earlier slide. Dynein is a motor protein. So with the help of ATP, the dynein would attach to the nearby microtubule pairs, and when they pull with the help of ATP, it changes the shape of the dynein and pulls on it, and that's going to cause the entire structure to wave back and forth the way a cilium or flagellum would overall. So the cilia movements alternate between power stroke and recovery strokes, which we saw in the prior image. And the flagella are going to move slightly different. They're going to have more of a uniform waving back and forth rather than the recovery stroke, but overall the structure is the same, and the dynein is responsible, the motor protein, for pulling on those microtubules to get them to flex back and forth. Here's a animation showing that 9+2 structure with the motor protein, so let's take a look at it.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Now we're going to look at microvilli. And the microvilli are these other structures. They're finger-like extensions of the plasma membrane, and they project from the surface of select cells. Examples here include the cells that line the small intestine and the kidney tubule cells. We've seen kidney tubule cells before in the laboratory, and overall what this does is going to increase surface area. So for cells that are having to involved in absorption of materials, it helps to have more surface area across which you can absorb. So that's the role of these microvilli. And the microvillus is singular, so you can see in your diagram here, and then we zoom in and they give us an electron micrograph here. It looks like a scanning electron micrograph because it's 3-dimensional. And there they label a microvillus, which is singular. And now these extensions, basically it's a plasma membrane that extends outward, and what provides structure for this is actin filaments. So those actin filaments help provide a stiffening of these projections to give them that internal support they need. And again, overall, anytime you have a situation where you have to absorb things, then it would help to have more surface area there. So, looking at a question here regarding what we covered, the presence of which of the following cytoskeletal elements allows cilia with the ability to move and bend? Is it A, intermediate filaments? Is it B, microtubules? C, microfilaments? Or D, actin filaments?